So, hello and welcome to today's webinar, Improving Preclinical Milestone Predictions Demonstrated on nQuery. So today's webinar will hopefully give you an idea of how to do milestone predictions using nQuery, but also the general concepts as well. And then what kind of improvements you can get in terms of making milestone predictions when moving from the pre-trial scenario, the planning scenario, to using interim data, the real data that comes in to help make better milestone predictions, whether that's for enrollment or for events in the case of a time to event / survival analysis. Before we get started, just a few frequently asked questions. Is this webinar being recorded? Yes. The recording of this webinar alongside the slides will be sent after this webinar via email. In terms of questions or technical issues you may have, feel free to use the questions tab on the right-hand side of your webinar software. I'll try to get to some of the questions at the end, time permitting, but any I don't get to, I'll make sure to answer via email after the webinar is complete, either later today or early tomorrow. Okay, so I think that covers everything we need to go through today. So let's talk about today's webinar, Improving Pre-trial Milestone Predictions Using Interim Data to Improve Your Study Length Predictions. And so that's really kind of the core of what we're talking about here. Like, when we talk about nQuery, usually we're talking about sample size calculations. We're talking about calculating how many people you need in your study. Well, the next obvious question that comes after answering that question is, well, how long is it actually going to take for me to achieve that goal? And of course, that's very important from both the trialist's point of view, but also from the sponsor's point of view and from patients' point of view as well, in terms of when is this treatment going to become available to the wider population. And of course, there's very famous examples recently with the COVID-19 vaccine trials where the speed of that trial ended up being important in terms of the turnaround that we've seen over the last year and a half since then. Before we get started, let me introduce myself. My name is Ronan Fitzpatrick. I'm the head of statistics here at nQuery. I've been the nQuery lead researcher since nQuery Interim 3.0, so that's about 7 or 8 years ago at this point. And obviously, we've had a lot of change then, adding in a Bayesian sample size, adaptive sample size, and most recently, milestone prediction using interim data or summary data. I've given webinars and talks at places like the FDA, JSM, and I think for the rest of this year, we're hoping to make places like JSM and the PSI conference. So keep an eye out for our appearances at those conferences. So in terms of today's agenda, we're going to just start with an overview of milestone prediction as a concept. What is it? Why is it important? And what value does it bring? And then we're going to talk about the two different scenarios where a milestone prediction will be likely to occur. One is pre-trial, almost kind of a natural extension of the sample size calculation, where we want to know pre-trial roughly how long is the study going to take, and we can do that in a kind of formal statistical manner using things like simulation rather than just relying kind of like on an ad hoc calculation. And then interim prediction is where we have access to interim data, perhaps an interim analysis from a group sequential design, perhaps just because we're enrolling data as we go along live. But regardless, we have data available to us as the trial is going, and we want to improve the predictions that we made before the trial, or maybe that didn't happen at all, and get a better idea of whether our trial is still on track, for lack of a better term, at the time of the interim analysis. And then finally, just some time for some minor discussion and conclusions. Just to mention, today's webinar will be demonstrated on nQuery, your complete solution for optimizing clinical trial design. nQuery obviously covers a wide variety of different clinical trials, from preclinical research, early phase, phase 1, phase 2 type designs, but obviously with its strength being in confirmatory phase 3 designs and the analyses that are used in that context. But I suppose just to mention today that nQuery is primarily about study design in the context of sample size determination. This is kind of the latest feature that we added late last year, nQuery Predict, which is related to our expert tier of the software, and that's kind of a natural extension of the sample size calculations that we already have, and hopefully provides additional value, as you'll see today. nQuery is obviously widely used in the pharmaceutical and biotechnology industry, with 88% of organizations who had clinical trials approved by the FDA having a license for nQuery, and you can see some of those in this slide and some of the reviews that we've had. Okay, so I think hopefully just covers the basic marketing requirements for nQuery there. So let's get into the meat of today's webinar. Firstly, kind of giving a description of what milestone prediction is and why it's important, and then how it's slightly different for the case of enrollment or sample size prediction and for event prediction in the context of a survival analysis. So in terms of milestone prediction, As I mentioned, it's kind of an extension of sample size determination, which is obviously the calculation of how many people you need in your study. The Goldilocks problem of you want, not too few subjects, but not too many. In the you don't want too few to avoid incorrect inferences, and you don't want too many to avoid additional costs and exposing patients unnecessarily to an unproven therapeutic. And I think the important thing to note here is that sample size determination is primarily focused on statistical success criterions, most commonly statistical power, but sometimes interval width or cost-based or decision theoretic approaches as well. I think it's just worth noting also that like if milestone prediction is downstream of sample size determination is itself very much downstream of the big choices made when designing a trial, whether that's regards like which groups are going to be compared, which statistical tools are going to be used. All of those go into the sample size justification, ideally. Now, obviously, there are approximations that work more or less fine for most cases, but from our point of view, SSD is itself kind of further down the chain, and milestone prediction is kind of one link lower than it. Milestone prediction, of course, then is focused on how long you expect it to take for you to reach some key target, like, for example, reaching the target sample size you calculated during your sample size determination, or the target number of events that you specified if you were doing a survival analysis. And these milestone targets might be related to the study end very commonly, or it might be like how long do I expect it to take until we reach our next interim analysis in the context of, say, doing a group sequential design. And so I think the important thing to note here is that milestone prediction is focused on a pragmatic issue, which is like the length of time it will take for this thing to happen, and isn't as worried about the statistical concerns of like what's our hazard ratio right now, what is our power right now, conditional power, anything like that. It's purely focused on that idea of like we need to reach a certain goal, a certain sample size. That's what we put into our protocol, and that's what we agreed with the regulator a priori in our protocol. Now let's talk about what that practically means in terms of how long this study is going to take. And of course, sometimes your trial will be ahead of schedule because things end up going better than expected. For example, those COVID-19 vaccine trials ended up going quicker than expected because there was a large amount of COVID-19 in the regions that were selected for patient selection, which ended up being useful for those trials. So obviously, not great for the people who are in those jurisdictions. But of course, we're generally more concerned with the opposite problem, which is when a trial is off schedule, when it's behind schedule, and that's an incredibly common issue in most clinical trials, where 80% of trials end up being delayed, and 85% fail to even reach their recruitment goals in the first place. So these are obviously significant issues that affect real trials, and therefore having an early warning system that would let you know that that's something that you should be concerned about based on where things have gone so far is important. And it's, if you look at the pattern of accrual in real trials, what you will often see is a ramping up over time. So basically, things take time to open sites, to open hospitals, to get access to patients. And then what happens is that, as they come towards the end of the planned accrual period, you see a very rapid increase in accrual to try and get to that. And that just reflects the constraints of actually trying to recruit real people from real sites, real hospitals. But you obviously would ideally try to avoid that. I think it's also worth noting here that when we talk about milestone prediction today, we're kind of going to talk about like an abstract statistical sense, where we're effectively assuming there's some kind of statistical process, some underlying statistical model that's generating these enrollments or generating these survival times. But in the case of enrollment, we know that under the hood, there's a lot of very important work being done by real scientists and trialists and people involved in the logistics of the trial. So just to say that we're not worrying too much today about the realities of that, whether that be the effect of screening, the effect of the length of time in terms of getting a site started up, or in terms of like dropout, in terms of like dropping out in terms of enrollment between like the first assignment and then actually being scheduled for your visits and stuff like that. So from our point of view, we're going to treat it like an abstract problem where there's a known statistical process kind of generating subjects without too much concern necessarily about those, like all those complicated steps. The one level that we will worry about is the difference between treating enrollment as if it's coming from kind of a global process versus treating it as it comes from a site level process. But in terms of what we're treating here, it's kind of as a statistical process, not as a kind of multi-stage model or Markov type process. Just to, I suppose, note that up front. And so that's probably reflected on this slide here in terms of what we're talking about when we talk about enrollment prediction. And it's basically just like, how long do we think it will take for us to reach our enrollment milestone? Like, for example, how long will it take us to recruit the number of subjects that we plan for from our sample size determination? And this is obviously a milestone that's relevant for pretty much any clinical trial that you can think of. Nearly every clinical trial will have some a priori target for how many people they want in their trial. And this is often referred to as a recruitment milestone or an accrual milestone or a sample size milestone, as well as being like an enrollment. So these are all interchangeable terms with each other. They're all describing the same thing. And as I mentioned, there are like multiple levels and approaches that you could do to look at enrollment prediction. Like if you really want to get into the weeds and deal with things like screening and like the actual process of getting a site up and running, you can go down to that level. There are other like specialized software for that, which are more really focused on the logistical side rather than the statistical modeling side. And even within the statistical modeling side, you can look at like, are we going to treat this as a global process, a regional process, a site level process, and do we treat each of those as being specified independently by the person distinctly by themselves, or do we use like a statistical model, which a multi-level statistical model, which kind of tries to estimate using random effects, kind of roughly where they should be based on the global process or based on the information available to us. So there's a lot of different ways you could go with this problem. I suppose, the big advantage that you would hope to get from doing an enrollment prediction is that it would give you important context for your major trial logistical decisions, like, particularly, of course, in the case where the trial is behind schedule, where the recruitment is behind where you need it to be or where you were initially planning to be. And that would then give you the ideas of what you need to do to improve enrollment at that point, whether that be adding additional sites or taking additional action on a particular site. If you look at the different sites, see the ones underperforming, relatively speaking, or other decisions that might be required, like changes to the screening program are unlikely, but perhaps might be considered if things are particularly dire. But the main point here is that if you're doing this before the trial, you're getting an idea of what might happen using our sensitivity analysis and trying different scenarios. And if you're using interim data in particular, then now you're getting a snapshot of what's likely to happen going forward based on what's happened so far. And then you can hopefully accurately model what's going to happen, both using the information available to you and perhaps using information and context from prior and other trials. Just to mention that when we're talking about prediction, there's a whole suite of different ways that you could do this. You could just use a kind of a classic linear fit, that just like take the recruitment so far and just draw a straight line through it, or fit a polynomial to it, and then just kind of take that as a kind of a fixed parameter estimate. There are kind of piecewise models that you can use, like the Poisson. You can slot things into an equation, for example. The ones we're going to focus on today, because they're the ones that use an nQuery, are based on simulation, which is obviously has the huge advantage of being very, very flexible and allowing you to deal with a wide variety of different models and scenarios pretty trivially compared to explicit formulas from models or and such. There are machine learning and other approaches, and obviously you could integrate things like the effect of cost and stuff, or optimization over cost as well. But today we're focusing on that, we have a target time or length of time we want to take for this study to complete enrollment, complete recruitment, and we want to use a statistical model to simulate from to see how we're doing in terms of that target. The other major area where nQuery focuses on in terms of predictions is on survival event prediction. So this is the case that in a time to event or survival analysis, we're focused on the inference about how long it takes on average for someone to have an important clinical event, whether that be death or disease progression or some other critical outcome that's of importance to the trialist at the time. Obviously, in the case of oncology, the two famous ones are overall survival, or OS, and progression-free survival, PFS. Which obviously deals with the case where those who aren't OS PFS are those who either died or progressed in their cancer, for example, for both of those endpoints. And of course, the important thing to note there is that we don't know a priori how long on average people take to have the event, because that's obviously the statistical question that we're trying to answer, the one we're trying to make inferences about, which means that you have a much higher degree of uncertainty regarding this outcome when event is going to happen. And when we're talking about survival trials, we're usually talking about there being a target number of events. That we're talking about an event-driven trial where we have a specified number of events that we got from, for example, a sample size determination, which includes the number of events as one of its main outputs. We're talking about how long it's going to take to get to that number of events. That's the key piece of information from the point of view of the trial. And the sample size, therefore, really in this case, if we're worried about it, is really just chosen to give us how long, give us the number of events required, how long the sample size is really the number of subjects required to get to that number of events, given some different constraints, like what we expect the hazard rates to be, how long we want to recruit subjects for, how long we expect the study to be in total. And these all feed into the sample size calculation. But just important to note that the events is the target in a survival trial, not the sample size typically. So it's like we're waiting for 200 events to occur. The sample size could be 400, could be 4,000. It doesn't really matter. All that matters is when that target event is reached. And that's why it's important that we're modeling this in the case of a survival analysis trial. I think the big thing that brings in is that if we're talking about an event milestone, then we're not only going to need to model the survival process by using, like generating from a log, like from a exponential distribution or Weibull distribution or similar, or from other Cox regression type models as well. But we also will often need to look at also doing the accrual, the enrollment process, and also perhaps many other competing processes as well. So if we're talking about the accrual or enrollment process, that's really just bringing us right back to this, right? So as part of doing an event prediction, we're likely, unless the enrollment process is already complete at the time of an interim prediction, we're likely to have to model this enrollment prediction at the same time, because obviously the time to event in terms of compared to the start of the trial will depend when a subject actually entered the trial, right? So if a subject enters at the very start of the trial, then obviously we don't expect their time relative to study start to be the same as from someone who entered the trial 12 months later. For example, that person who entered the start could very well have already had the event well before the second person, a 12-month entry. And in terms of competing processes, this could be anything relating to dropout, censoring, cure processes, competing events, anything that basically means that subject will not be available to make inference about from the purposes of the power calculation, from the purposes of the survival analysis model. So it's important to note that really, what that other event doesn't really matter unless you're planning, for example, in the case of a cure model, to include those people somehow in the final inference. If you're not planning to use someone who's having a competing event or dropout in the final inference, like the final effect size, your hazard ratio, then really all of these processes are effectively treated the same, in the sense that they're just processes that stop you from having the event of interest. And basically that's what we're actually interested in from a practical point of view in a clinical trial and survival analysis. It's important to note that I suppose that events prediction models will tend to be much more flexible, which kind of reflects the fact that sample size calculations tend to be more flexible, and analytic and simulation approaches are available for this type of problem. And implicitly, like many of the sample size calculations that you use in survival analysis, basically already include a component that would allow you to calculate the length of the accrual period and the total length of the study for you independently. For example, in nQuery, if you look at the table STT22, which deals with the case of where we only know the recruitment rate and where we want to then find the accrual rate and the final study length, that actually comes up explicitly. And so there's a variety of different models that you could use, like exponential, Weibull, log normal, etc. And also the same models are often useful for modeling processes like dropout, for example. And so we need to fit our process, we need to get a good, accurate estimate, preferably, of what the survival model would be. Or if we choose a survival model, we want to fit to the current data that exists, the current length of time it took for each subject to have the event or not have the event, in the case of people who are still alive. But we also need to have a model that we can generate future survival times from, whether that be people who are currently in the study and could have the event in the future, but also subjects who will be recruited into the future and then will have their own simulated survival time. And of course, we're dealing with a lot of different constraints here. For example, if we're dealing with survival data, do we treat this as coming from a global process? If in the case that we have blinded data, do we treat this as with each group, do we know the hazard ratio and the independent hazard rates if we have unblinded data, and do we model that such? It'll really depend on what kind of data is available to you. But we can integrate all of those constraints into our modeling, into our problem. That's also true for enrollment, but it's just much more of a consideration for survival data, where obviously ideally we would know what the hazard rates are in each group, but obviously we don't generally know that if we're talking about someone who's not on the data monitoring committee, for example. And as mentioned here, like enrollment modeling is a intrinsic part of the problem, unless you happen to have already finished enrollment at the time that you choose to do this prediction modeling. So this is a very similar set of options available for the case of survival, event prediction compared to enrollment prediction, like from parametric modeling, simulation, which is what we'll focus on today. There's model selection approaches and machine learning. And if you're talking about like more modern problems like proportional, non-proportional hazards and similar, or time varying covariates, then there's a variety of different independent approaches for those things as well. And if you're talking about trying to get the best fit or estimate for what your model should be doing, there are also Bayesian approaches or expectation maximization approaches, which offer alternatives for kind of hopefully trying to either use independent data to improve your estimate, or to give you kind of decent estimates of an unblinded effect if you only have blinded data available to you in the case of EM. So hopefully that gives everyone a good taste of what we're talking about today. The rest of this webinar will have some brief introduction on the unique problems of doing these types of predictions at different points in the study's lifetime, but mostly we're focusing on like what does this practically look like, what do these choices look like in a software such as nQuery, when we actually want to get down to brass tacks and actually do these predictions. Like briefly, I suppose on pre-trial milestone prediction, like we're trying to do this prediction of how long our study is going to take to either finish or reach the next interim analysis before the study starts. So we want to know roughly how long is it going to take for this study to take, like how long is this study going to take to reach its goal. And we're obviously, as I mentioned here, focused on the practical aspect of study length, independently really of the power and the sample size determination problem. All we're taking is we have this sample size or events we need to reach. How long is that going to take based on our pre-trial assumptions? For our enrollment prediction, all we really need for enrollment prediction is some reasonable estimate of how we think the accrual process will be. Usually that can be summarized down to a single accrual rate, and you can use a simple model like the Poisson, or if you're looking a little more complex, maybe piecewise Poisson. Potentially you could look at like digging into the site level stuff immediately, but today we'll probably just focus on just an accrual rate and a fairly simple model. Of course, where does the information come from? You're likely to use information from previous trials or from expert elicitation or from people down in the field to kind of get an estimate of how long, what that accrual rate's going to be. And then a simple accrual rate and a simple sample size target can usually get you a reasonable estimate of time pretty trivially. For survival event, things are a bit more complicated because you need to have access to information or ideas of what you think the hazard rates are going to be in each group, what dropout rate might or might not exist, and then the expected length of your accrual period and any like differences you expect in terms of the accrual process itself, like uniform versus non-uniform, and then also your study length. You'll probably have an idea of that from your sample size calculation. So I think the big thing here is that that is a lot of additional information, but it's all information that you're likely to have already derived or got estimates for the purposes of your sample size determination. And so from that purpose, the sample size determination for this case and for events calculation and for event prediction are very, very similar. And indeed, certain statistical models that have been proposed for sample size determination for survival analysis can actually be used explicitly for this purpose. One of the well-known survival sample size methods is from Lakatos, based on a non-stationary Markov process, and it's not actually that difficult to rewire that to generate expected study length from that type of model. So that's just a model where you have, everyone's live and available to have the event initially, and then you create a transition matrix for the different types of things that can happen and to deal with things like accrual, and then you just kind of run that, and you can use that quite easily to get an event. But of course, that is a very flexible approach, but it's not as flexible as simulation. Because simulation can then go into lots of other distribution types and changing the transition matrix even more so, and a lot of other things that are just very easy to do in simulation, but slightly more difficult to mark up. But like the Lakatos approach is very, very flexible if you're interested in a analytic approach that will give you the same result each time. And for that reason, you can also, to some extent, choose to treat this milestone prediction for survival as a sensitivity analysis for your sample size calculation. Particularly the calculation of, like, I said the study could take 40 months. Now it's going to take 42 months because of a change or sensitivity analysis that we, like, something we choose to change for sensitivity analysis, and that all kind of makes sense, right? And so what I'm going to do here is I'm going to use an example from the nQuery manual using the information that was provided for a sample size calculation. So this is based on a real trial, but some of the facts have been changed around. So there's a real trial where the summary data was available, and we had access to the actual underlying data as well. And we just created an artificial version of that trial with some of the facts changed slightly, but broadly the same trends as we'll see in the interim data, which is kind of like simulated from the kind of summary data and real data we saw there, is kind of just derived to kind of be somewhat self-similar to that real data, without obviously exposing real data to various usage issues. So in this case, we had a case where they had a hazard ratio of around 0.715, and for the power that they wanted at the one-sided 0.025 level, they needed 374 events. And based on hazard rates in each group of 0.065 and 0.09 for treatment and control respectively, they said we need 40 months, assuming uniform accrual of 460 subjects over 30 months. And what we can do using nQuery Predict is first verify that this prediction is accurate of 40 months. Obviously, we'd expect trivially for that to be true, but also then explore perhaps a scenario where the assumptions used here aren't true and what effect that would have had on the expected length of this study. So this is a survival analysis study. How long would it take? And obviously, we're dealing not only with modeling the survival process, but also the recruitment process. And of course, we could also model things like dropout if we wanted, but we'll keep things simple today and avoid dealing with dropout for now. So I think in terms of nQuery, the first thing I'll just do is make sure that the sample size calculation, we can verify that. So this is kind of like, naturally before we'd be doing a milestone prediction, we would usually have done a sample size calculation. And so we can see 30 months is the accrual period, 40 months is the maximum length of follow-up expected. We had our exponential parameters here of 0.065 and 0.0909. And if you're interested, you can also put these into the side table to get the kind of median survival. So we're talking here about median survival of around 10.66 months in the treatment group and then 7.625 months in the control group, with a hazard ratio of around 0.715. We'll click transfer to move those in the main table, and then we'll set the power to 90%, obviously the more commonly used. And we'll see here that we get a sample size per group of 230, or the 460 in total that we have talked about in the slide, and 374 events in total. So this is a very trivial sample size calculation, nothing too complicated. I'd say this isn't exactly the same as the one from which this trial we're kind of basing the kind of trends off, but it's quite close to it. And so, this is our sample size calculation. We'd assume that usually exists. And so now we want to do a pre-trial milestone prediction. We want to see, verify effectively up front, like if we use simulation, do we find that given these hazard rates and this accrual process, will we end up with a study length of around 40 months? And so to do this, we open up nQuery Predict, which can be done from a variety of different options. So if you go to the home screen, the default screen when you open nQuery, you can go to click create prediction here. And since we're doing a pre-trial prediction, there's no interim data here. So in this screen, we can just click skip to open up a fresh prediction workspace, which doesn't contain any data, which means we're limited to using summary data here in the input type. But you can also select this from the new prediction option here in the toolbar, or we can click from the predict file menu, and then we go to prediction with fixed parameters here. And those will open an empty workspace where for now the only option is to use summary data. Now we can always add data at a later date by selecting the add datasete option from either here or from the predict menu, and that's when we can open data and turn this into one where we can do interim predictions. But for now we don't need that, so we'll ignore that for now. So what we want here is to use unblinded events, because obviously we have the we have the two rates here, so we can use the data like properly, accurately for each group rather than pretending there's some kind of global process. And the enrollment status is ongoing here because we're going to choose to, set this at the start of the trial. I suppose it's important to note here that if we're doing a summary data prediction, we can use summary data for interim predictions as well. And if we were doing that, then we'd be entering like how many people have been recruited at the summary data, like at the current interim data, say like 100 in each group, and then 50 people had an event. And we fill in this fully and we click next. But for the case of a pre-trial prediction, we can actually just set these all to 0. And so if we set these all to 0, implicitly we're setting this to a pre-trial prediction, right? So this is effectively the same as saying is that no recruitment has happened yet, no events have happened yet, and the current calendar time is 0 compared to the study start. So this is a pre-trial prediction. And of course, because there's no prior information available, nQuery won't provide any really amazing defaults here. It will kind of just default to things that kind of make sense. And so what we want here really is to set the target sample size to 460 as per the original slide. And then we need to set what we think the recruitment rate is going to be going forward. And if we're assuming a constant uniform accrual rate, that's pretty trivial. All we need to do is take the expected sample size, 460, and divide that by the expected length of the accrual period, in this case 30 months. And so we can just go to, say, the assistance menu and click the Windows calculator. We can take 460, we can divide that by 30, and we get a accrual rate of around 15.333 subjects per month. And we'll replace the accrual rate going forward with that value there. So here we're affecting what the future Poisson uniform accrual rate is going to be. We click next, and we move on to the event and dropout information step. So, the rough outline is, step 1, we're selecting what type of prediction we want. Step 2, we're specifying the type of data that's available, whether that's the data sets that exist for interim data, or for summary data, just the inputs that we have. We then talk about the accrual process, if it exists. If enrollment's finished already, this will be mostly just providing you information that you already know. And event and dropout information is when we're going to specify the survival process and model. And so we know that the target number of events is actually 374. And we're going to assume here an exponential model because the exponential model is unconditional. It doesn't, there's no change in the rate depending what's happened in previous subjects, and so it's an easy model to generate from even in a pre-trial stage. Other models you can do it. There are reasonable ways to do it for other models, which we're adding. We plan to add over time in our biannual updates, but for now we're limited to exponential here, and so we're going to use the values directly from this sample size calculation here. And so we'll just take this by copy and pasting the hazard ratio here. And we'll take the control rate of 0.0909. And so you can see here that the treatment hazard rate automatically calculates the update to be the same as from the original calculation. And of course, if you change any one of these 3, the other one will automatically update to ensure that there's consistency across these. We're not worried too much about dropout today, so we're going to set the dropout to 0 instead of these default values of 0.01. And so now we've set up our predictions so that it's basically giving the same assumptions as STT1 here, this sample size calculation here. Implicitly, there's no dropout here because there's no row for it in the main table. STT2 would be a table that would have dropout in it, and in that case, we would then deal with the dropout model process here. Once we've entered our event and dropout information, we can click next, or we can click this button up here if we want, and that takes us to the simulation controls. So this is just the final step in a simulation in nQuery for enrollment or event prediction. And for the milestone prediction, we want to specify how many simulations we want. The refresh frequency is just for how, when we're getting updates on the trial, on the simulations we're going along, it'll update occasionally to give us the best estimate so far. And then we can specify what type of output options we want. And in this case, we have 2 options, which is to say summary statistics for every simulation run, which just means that we'll get a table that for every simulation, we'll get an idea of how that simulation did in terms of how long it took for that simulation to complete and how many people were recruited, etc. And then this option here allows us to save one or more individual simulations, so we can get the actual simulated event times, dropout times, recruitment times as needed. And just down here, we will get a percentiles table in our result that gives us like the 5th and 95th percentile over all of our simulations, as well as 25th, 75th, and 50th percentile. 50 percentile would be kind of busy if you want the median result across all your simulations. That's what this is here. So we click run here, and you see the simulation is running and dropout isn't used here, so it's empty. And we can see here, as we're updating, we're very close to the actual expected average study duration of 40 months. And when you get into nQuery Predict, there's a variety of different options to explore your results. There are plots, which hopefully give you a decent visual idea of not only like what was the actual trajectory, but also the level of uncertainty across all of the simulations. This 95% prediction interval is effectively, 95% of the simulations had a sample size at any given time between the two values specified here. So you can see it at a time of around 25.898. The simulations, 95% of them had a sample size recruited at that point of between 360 and 438, with an average of around 398 at that point. So around 400 people recruited at that point in time, around 26 months in. And then the events prediction, of course, is integrating the effect of the enrollment prediction as well as the survival process in this case, and obviously the dropout process if that existed. And you can see you get a slightly more complex case here, because obviously instead of just talking about a fairly simple Poisson model where we'd expect a linear increase in sample size over time, we're dealing with all of that interacting with the two exponential processes that are happening at different rates in each group, and we get something that looks something like this. Important to note here that in terms of the events versus time plot, that we're talking about when these events occurred relative to study start. This is not a Kaplan-Meier type curve where obviously you're adjusting to when the person entered into the study. This is purely point 0 is when the study started, and this point here is around 40 months after the study started. This is not really this isn't self-adjusting Kaplan-Meier style for entry time. This is purely how many events occurred relative to the study start time. If you want more detailed information, you can use the simulation summary report. And this just provides a very detailed analysis of what went into the trial, like what went into the simulation. So pretty much every single input is provided on this left-hand column here. And then on the right, we get a overall summary of our results, which provides us not only with information on what happened in total, like the average study duration here, you can see is around 39.78 for 3 months, which is very close, of course, to our expected study time from our sample size calculation of 40 months. But also that in 100% of these simulations, the target number of events of 374 was reached. Just a small note here that for event prediction, it's all about the events. If you happen to have a study where the events target was reached faster than expected and before the recruitment process was actually finished, nQuery Predict, this feature will actually cut off the event, like the sample size, the enrollment process early, and basically say, well, we don't need to recruit more people because we reached the target number of events. And you can also do things like dealing with a fixed follow-up where subjects are, like, we're assuming by default usually that subjects are followed up until the end of the study, at which point when the target event is reached, and then the remaining subjects are censored. But we do also have an option in the accrual option step where you can set a fixed follow-up process for the case if you have interim data. This isn't integrated for the pre-trial assumption, but if you want to do it for the other case, you can. And so you can see from the simulation summary that not only do we get information about the overall trial, but also on what happened in each group. And you can see there's around 197.5 events happening in the control group on average, compared to 176.4 in the treatment group. And then you can get the percentiles of what roughly happens. So you can see that, for example, for study duration, 5% of the simulations had a study duration less than 37.175 months, and then 5% had a study duration above 42.4457, and then 25%, 75%. Like the interquartile range is basically around 38.7 up to 40.86, and our median study duration was around 39.7646. And the accrual duration, you can see, is also centered around 30 months as expected, so that seems to be accurate. And so what you're really getting here is that this more or less verifies that what we were expecting from our sample size calculation is still true. But, of course, as mentioned, things often aren't what they seem. Things often go worse than expected in terms of like how long things actually take. And what I'm going to do here is going to cheat a little bit, give you a little preview of the interim prediction. I'm going to go through this in detail in the next step, but just to say that when we use the actual data that this sample size calculation and the pre-trial prediction is based upon, this is what the interim data around basically when half the target events has arrived, which is a very obvious a time for an interim analysis to happen if you're doing group design. So if basically after 187 of the 374 target events has occurred, a hypothetical interim analysis has happened. You're now doing a milestone prediction, and this is what the data that we've simulated from or simulated actually gives the hazard rates of basically being 0.779 and 0.0547, which you'll note are both lower than expected. Though you'll note that the hazard ratio here is actually lower than the original target hazard ratio, which means that even though our event rates are slower, we do have a better hazard ratio, which means that in terms of power, we might actually be okay. But what I'm going to do here is going to cheat a little bit. And what I'm actually going to do is going to take these values from the actual simulated interim data, and I'm going to feed them back into this step here for our pre-trial prediction. I'm going to see, like, if we had known this a priori, what the truth in inverted commas was, what would that have looked like in terms of the expected length of this trial. And you'll note here that, of course, these two rates are lower than the two rates that we originally had. 0.77 for the treatment rate is significantly lower than 0.09. And that means that, of course, a priori, we would expect this trial to take longer because obviously events are coming in slower than expected. The event hazard rate is lower than expected. And so I've just changed the options to this step, and you can always go back to any step you want and make a change. And as long as you go click next when you finish your edits, and you move over to simulation controls, then you'll be able to rerun this. So remember, originally we expected the study duration to be, just below 40 months. We were looking at 39.8 or so. And you could see that if the true rates were what we got from the interim data that we're going to talk about in a moment, that we simulated for this trial, then the length of the study expected, the average study length actually now increases to over 43 months. So that's a delay of around 3.2 months. Or 3 to 3.5, depending on if you're going to use the original 40 or you're going to use the simulated value. But either way, a somewhat of interest delay. Probably not one that would cause a huge ruction or change to the trial, because likely whatever changes you would make by the time they'd actually have an effect, you would be past the point at which they would actually make much of a difference. So this is probably one of those cases where you just have to take this information, provide this to the relative stakeholders, and hopefully create the correct expectation in terms of when this trial is likely to end based on the information available thus far. But that just gives you a flavor of the kind of things that you could explore. Of course, there's lots of other things you could explore. You could change the expected enrollment rate, and then in future, you could also look at other models as well. So that's pre-trial prediction. But the obvious point to make is that if you're doing a pre-trial prediction, you're obviously using information that it's obviously probably from prior studies or based on expert elicitation or based on domain knowledge. But obviously real trials, real information will differ from that. If we knew what the trial was going to do a priori, then we wouldn't need to do the clinical trial in the first place, obviously. So if we then are using interim prediction, if we're using the real data that becomes available as we do our trial, as we're going through it, then why wouldn't we use that information? That's one of the many other reasons why we do this, to improve our milestone prediction. So if we're doing interim analysis in a group structure design, for example, we're already looking at the data, or at least the data monitoring committee is looking at the data. And one additional piece of information that they might provide is not only is like, here's, are we choosing to end the trial early or not, but perhaps also roughly an estimate of like how long the trial is going to take based on the current trajectory to complete. And so, yes, so we have interim data. We're going to use that to improve our prediction for these key trial milestones, whether that be enrollment or events. This kind of gives you a snapshot of if the trial is on schedule and we want to compare to really our pre-trial assumptions, or basically even our any kind of prior prediction of how long this trial is going to take, whether from a sample size determination or from a pre-trial prediction, as we just showed there a second ago. And of course, it goes without saying that this is information that will be highly valued not only by the people doing the trial on the ground, but also, of course, from sponsors. As I say, if a trial is ahead of schedule, not a big deal. But if a trial is behind schedule, or there's a potentiality that, based on the current constraints, it's unlikely to reach its enrollment or event goal, that's obviously critical information that would be very important to know. And I think the important thing to note about interim milestone prediction is that the tools that are available, and nQuery provides a subset of the tools that could exist in totality, should adapt and should be able to adapt to the information available to a given stakeholder while the trial is ongoing. So blinded data might be available to people who are involved in the trial itself, but unblinded data may only be available to the independent data monitoring committee if we're talking about doing survival analysis modeling, event modeling. If we're worried about the enrollment process, then we might have either, like, here's how people have recruited in total, but we might also have information on each site that's being used in the current trial, and we might have summary data versus actual data available to us. Like, we might have, here's what's happening in each site, or we might just have, here's what's happening in a broad sense. So do we have summary data, just like here's how people have been recruited over this length of time, or do we have, here's actual information on which every single subject arrived into the study? Do we have unblinded versus blinded data if we want to model survival as a global or per group process? Subject versus site data if we have interim data. Do we also have site level data which to improve our enrollment prediction? And so with nQuery, we actually now provide a couple of, like, those two simulated data sets that I referred to, called subject data.csv and site data.csv. These are actually provided now with the software. I'll show where in a moment, but also the example I showed previously, as referred to in the title, nQuery Predict example 6.2.7.5 scenario A, but also all of the other interim data predictions use this data set in the updated nQuery manual, which you can find by going to the software and clicking help, and then nQuery manual. And then that will open up the nQuery manual, which obviously here I've just opened up. I already had it open up on a page near to where the example, the first example we did is, and you can see here the sample size calculation, for example. And this provides a background not only on what milestone prediction is and the statistical models used, but also gives a huge number of different examples, worked examples from end to end on each of the different scenarios, whether that's unblinded data versus blinded data, site level data versus subject data, summary data versus actual interim data. All of these are covered in detail in this chapter. So if you're interested in seeing lots more examples of the software in action, I would very much recommend seeing the updated manual that's added. Note that this was added in the 9.11 patch. If you don't have the 9.11 patch, I would recommend keeping up to date with the latest version of nQuery by going to the update feature, which you can find here in your taskbar, or you can find it on our website. If you go to our website, you'll be able to find the latest version of the manual, and you should be able to find the data sets as well. But I will note that the data sets will be sent with the email after this webinar as well, I believe. So just to say that the interim data we're about to use was conducted when 50% of the total events required had occurred. Enrollment was 87% complete, so 402 out of the expected 460 subjects had been recruited. And if we're talking about the site level data, 118 out of the 127 sites planned had already opened. And we can talk about a load of different scenarios. I think today we'll focus just on the unblinded case, because that will allow us to match more closely with the expectations we had from the pre-trial assumptions. But just as a note here, there's lots of different scenarios we've explored, and I've done 2 previous webinars that have used nQuery Predict, albeit not necessarily using this data set in every case, but they've gone through a load of these other scenarios as well. So if I don't cover anything that you're interested in today, get in touch. We'll be happy to share that material and see if that might be more amenable to what you're looking for. For example, I believe I've mostly gone through blinded in previous webinars. So in terms of doing interim prediction in nQuery, like as I mentioned, I've already done an example here where I've kind of set this up, but let's kind of reset this and do this from scratch, kind of just make sure everyone's on the same page. To do this, we would usually go to create prediction here on the home screen, but we could use prediction with subject level data, or same option from the predict menu here. But let's go here and we go to the new prediction menu. And what we want to do here is we're using interim data. We want to use browse here in the new prediction window. And I've set it so the place where the data sets will be found in your installation folder is set as a default. We can change that from the options menu up here. So if you go to file and you go to options, you'll see there's a field here, default predict import folder, and you can change manually what this folder is. So if you have a folder you're planning to save your future files in, you can change that within the software. By default, it should set to your default documents folder. But as I said, I've set it here so that it goes to this folder here, which is, as I say, in your installation, which is obviously within Program Files 86, Statistical Solutions Limited, nQuery, Test Data folder, Predict. So if you go to this folder in your software, if you have the 9.11 patch, you'll be able to find these two data sets, and these are the two simulated data sets from a real trial with some minor changes made to ensure not completely the same, ensure they're not completely the same. But we click import then. I just selected multiple files there by holding down the control key. And we click import, and then that will move us into an nQuery prediction. And you can view the data sets by selecting them from the data field of the workspace on the left-hand side. And you can see here in our site level data, we have which region a site was in, the site ID or the site ID, SID. The cap is like the maximum number of subjects that could be recruited from a given site or hospital. The open time was the length, like the time at which a site opened for sites which were open already at the time of this interim prediction. The rate was the number of subjects recruited given the time at that point. So, number of subjects divided by the length of time that site has been open. And start and end here, they're not used too often. This is really just the window of time during a site could open for the unopened sites. And we look at the subject level data, and just a note here in the manual, I go through all of these data sets, these two data sets in detail and what each of these columns is. So if you want more detail, I would refer to the manual. We can see region and the site ID that each subject was recruited from is given here. So just a note here, in this site ID data, each row is a site, which may be open or not open. In the subject level data, this is the actual recruitment time and other information that occurred for each subject. And you can see we have the arrival time, that's when they entered relative to the study start. The follow-up time, which is how long they've been on the study, either until the current time, like the time of the last recruitment in this case, or until they had an event. So just to note here, current is your current status, where 0 means that you have not had the event at the current time, 1 means that you have already had the event, and -1 means that you've dropped out. And so that means that for people who have a 1, that means that this is how long they were under the study until they had the event of interest, until they died. And for -1, that means the length of time until they had dropped out of the study. Whereas for people who had 0, this is the length of time until they arrived at 3.55, and then they had 21 months of additional follow-up. And of course, for people who have current status, you would expect the sum of arrival and follow-up to be the same for all people who have 0 current status who have not had the event. A treatment is just a treatment group assignment, 1 for treatment group, 0 for control group. And that's basically our data set here. So in this case, we want once again to target an unblinded events prediction, but in this case, we want to use subject plus site data. We could obviously just use the subject data alone if we wanted to, but we're going to model the enrollment by looking at the site level data as well. And so we select subject plus site data, and enrollment status is obviously ongoing here because we've only recruited 402 out of the expected 460 subjects. When we have real interim data, we need to obviously select our subject and site level data sets independently. So this is the subject level data step 2. And then we need to select the fields that existed in that data set based on the headers given in the original data set. And then we need to select which ones of those corresponds to the required inputs for this prediction. So we need the arrival time, which in this case is the arrival field. We need the treatment ID, which is equal to treatment. And we can see here that we could set that the control group is equal to 1 or 0, but by default, it will go for the lowest value in that column. In this case, 0 equals control group, 1 equals treatment group. We need our status indicator. nQuery predict is kind of expecting 0 equals censored or slash available, 1 equals event, -1 equals dropout, but you can actually manually set it to whatever you want. So current is the current status, and you can see here that 1 is event, -1 is dropout, and 0 is censored. Censored is really available. Censored is kind of referring to the fact that if we were to end a trial at the current time, then these subjects would be censored because they obviously have not had the event or dropped out. But that's equivalent to saying these people will be available to have the event going forward, which means that we won't be simulating recruitment time for them, but we will be simulating a survival and dropout time for them. The time in study is how long someone's been on the study up until the current time. As I said, the definition depends on what their current status is, on the value of current, where if event or dropout, it's like how long did they had an event or dropout. If they're censored, then how long they were on the study until the current time. And then SID is the site ID. And then in this case, we have site ID and we need to select similar to the same fields, like which site ID we have. And of course, any site ID that's included in the site level data, in the subject level data needs to have a corresponding site ID in the site level data. Otherwise, you'll get an error later on in the process. We have the expected rate of recruitment in each site, the enrollment cap, the maximum number of subjects allowed in each site, the open time for sites that have opened, like when did they open relative to study start, and then the window of time from like the start and end time of the window during which an unopened site is allowed to open. The remaining steps are relatively similar to what we saw for the pre-trial assumption. So I'm going to not spend too much time on this screen, for example, because this is very similar to what we saw for the same step in pre-trial prediction. But the big difference here that this table has disappeared, and because we've added site level data, we actually have this accrual info /site tab here, which will be automatically open if we click next, but can also be manually selected by selecting these two tabs here. I'm setting target sample size to 460 because that's what we expect. And as I mentioned here, for the case where we have interim data, we also have the option to set a fixed period for each subject for survival analysis, which means that after a fixed period for a subject, they will be censored regardless of when they entered the study. Relatively rare, so we're going to stick with the more common event-driven analysis until the end of the study here, which means that everyone's going to be followed up until the end of the study when the target event is reached. So I don't really have time to go over the event like this table here, but basically we have a recruitment rate in each site, which by default is taken from the number of accruals that have actually happened in the site. So 11 divided by the length of time the site's been opened, which may or may not equal the planned accrual site as you include in your original data. So that's for when the number of accruals is greater than 0. But if the number of accruals is equal to 0 or the site is unopened, this will be taken directly from the data set that you provided from the site level data. You can also see that the enrollment cap can be edited for any open site. And you can also see that for unopened sites, which are at the very end of the table in every case, and you can also add sites if you want by increasing the total number of sites. So if I increase this to 128, you'll notice a empty row at the bottom here where we can add our own custom sites if we want. But let's replace down to what was in the data set by default. And you can see we can change the period of time during which any given site can open, as well as the accrual rate and the enrollment cap as for the open sites. So we're not constrained to having, what's happened before is going to happen in the future. We can change any site's individual rate and enrollment cap at this stage later on if we want. The event and dropout information is pretty much the same as we saw for the pre-trial case, except that these values of hazard ratio, the hazard rates for the event and dropout process, these are based on modeling, on fitting to the actual data that's provided, to the actual subject level data provided. And so you can see that this is the best fit if we assume 2 exponential processes for the control and treatment group. But you can also have access to the Weibull process. If you want to use a Weibull model, we have the Weibull fit here. And of course, the Weibull model is different from exponential because it doesn't assume a constant rate, but assumes that the rate can actually change over time. So if you want a more flexible model, that's not as constrained to that kind of constant event rate, hazard rate assumption, the Weibull is available for you. So these two are the same, and the dropout model is also the same. That's been fitted to exponential. We don't do Weibull for the dropout process. It's usually less important. And then basically we have the same options here at the setup stage, but we do have the option to output the information per site as well. And so you know that obviously the simulation is a little bit longer here than for the other case because we're using the interim data and we're providing outputting a lot additional data here. And we're obviously outputting 2 additional data sets as well compared to the original case. But you can see here that the average accrual duration is around 27 months, and it's about 43 months for the study duration. So recruitment was actually faster than expected based on the process thus far. And that's probably reflected in what you'll see is the real enrollment curve that you'll see here. You'll see here that it kind of goes up kind of linearly mostly, like a little bit of logistic tail here, but then was kind of tapering off, which probably indicates that they were kind of, in this trial at least, probably already in the process of winding down recruitment because they were ahead of schedule. But in this case, we can see that if the overall process had been correct, and we'd probably want to change this recruitment rate maybe to better reflect this, and scenarios in the manual chapters actually go through that explicitly. But you can see here that if we just use the kind of linear extrapolation, we'd get around 27 months in this case, so slightly ahead of schedule. Whereas the events actually happens a little later than expected, where we're ending up with a average study length of around 43 months. So 43.7 months, so around 3.8 months longer than expected, which isn't too bad. And you could obviously change the enrollment process to be more conservative based on that graph here, but if you actually go through that scenario, it doesn't make a huge difference, I would say, up front. But that scenario, as I said, is explored in the thing. And you can see here that there's also a dropout plot here, because we included a dropout as a process here, because dropout had occurred in the interim data. But you'll notice here this report is mostly broadly similar for the pre-trial predictions, so there's no point in really going through that in too much detail. You're getting the same ideas here effectively of the 5 to 95% in range for the percentiles for each of the predictions, the per group values. I think the main thing to take away is that the recruitment is slightly faster than expected, and the study duration, despite that, is longer than expected. And that's not surprising, of course, because we talked about the fact that this rate, the WHO hazard rates that we got by the best fit at this stage were obviously slightly lower than the event rates expected from the original sample size calculation here of 0.065 and 0.099, 0.09. That's gone down to 0.054 for the treatment group and 0.0779 for the control group. As I mentioned, if you want information per site, those are available from the tables down here at the bottom. And as I didn't mention it previously, but if you want what happened in each simulation individually on average, like how long did the first simulation take to reach the target number of events, it took 43.978. But in simulation 2, it took 43.305 to reach the study end time. But if we want the information that happened in the first simulation, you can find that directly here. Obviously, some of these values are taken directly from the dataset where they've already been recruited and already had the event. But for these later subjects, these are being fully simulated, both their arrival time, their event time, etc. And if you want per site information, either what happened on average in each site across all the simulations, or what happened in a given simulation for a given site. So here's site 101. You can see that it had 12 accruals in simulation 1, but on average, 101 had, on average, it recruited 12.069 across all simulations effectively. Then that information is also available. So hopefully that gives you an idea of what that would look like. And of course, we could explore what would the effect of having the Weibull model, what would, we could also look at a piecewise exponential model. I just to show that briefly, at the event and dropout process, if we increase the number of hazard pieces to greater than 1, then we can have a piecewise survival process, exponential process, and a variety of other different options like the fixed follow-up I talked about and other things. And of course, in future updates, we'll be continuing to add many more options for simulating different scenarios. And if there are different things that are not available at present in nQuery and that you believe should be available, please do let us know, and those will inform the types of things that we are planning to do. For example, in our next update, we're planning to have some fitting for piecewise exponential models. Perhaps more importantly, we're also planning to add piecewise Poisson models for the enrollment process. So I think we're pretty much at the end here, so I'm not going to have time to go through the questions here today, but I'll email any, I'll have a reply email to any questions that you have. And as I said, the slides and recording will be sent to you after alongside links to the data sets. But just to conclude very briefly here, milestone prediction can be very useful for understanding and perhaps adapting your trial if it's off schedule. We want to predict that study length based on the best information available. So before trial, based on the kind of things we're using our sample size calculation, for example, for survival trials, while the trial is ongoing, using the actual interim data, or if we don't have access to that, maybe just a summary data of what's happened so far. So we know that 460 like 300 events have happened in the first 30 months. We can also use that if we go back to the fixed parameters, the summary data approach. Instead of using the pre-trial information, we'd use the actual information in that case. It's basically the same concept though. The two most common milestones are enrollment / sample size and events for survival analysis. And event prediction, as kind of very obviously from today's webinar, it basically will often include enrollment modeling as a sub issue, like a sub process within it. Pre-trial prediction allows us to explore important counterfactuals, with event prediction often using effectively sample size determination as its base for defining what the inputs for that prediction should be pre-trial. And then interim prediction, obviously because it's using actual information from your specific trial, should be able to give you better updated prediction information about how long your trial is likely to take, and that can then inform decisions about what's needed to get your trial back on track if it happens to be behind schedule. Or if things are ahead of schedule, you can take a glass of champagne and enjoy yourself, I suppose. So finally, I just want to thank you all for attending. If you have any questions today, you can email us at info@statsolves.com. So if you have any questions you haven't used today in the webinar software, please do feel free to get us. And I say, things like suggestions about things that we could be doing in future updates, very, very much valued, whether on this particular topic or any other sample size calculations that we could have available. Very, very much valued, very more important in terms of our prioritization index. And if you want further information on today's webinar, and the webinar will be up online at the statsolves.com website. But if you want any additional information, you can also go to statsolves.com. Do note that if you either don't currently have a version of nQuery, or you don't currently have access to the expert license under which Predict is currently found, you can try both of those for free depending on your level at statsolves.com/trial. You can start a 14-day trial free of charge. There's no commitment, credit card, etc. All we need is your email to send you a link where you can actually try this feature in browser without having to install anything. So it's a very easy, costless way for you to try out any of the features that are of interest, either in the wider software or specifically today, depending on your current, whether you have the software or given license level of the software. If you want access to our previous webinars or any training, etc., you can go to statsolves.com/start. And all the references used for today's presentation are at the end of the slides, which will be sent to you after today. As I mentioned, I think I've run slightly over time. I apologize for that. But once again, I want to thank you so much for attending today. I hope you have a very pleasant day. And as I say, if you have any questions, email us at info@statsolves.com. So once again, thank you so much for attending, and I'll talk to you soon. Goodbye.